Hello, everyone. I'm Chenwen Wang, currently a postdoc at the Greenberg Lab, Harvard University. Today, I'm presenting our study, STRAT-STATES, State-Based Visual Analysis of Disease Progression. This is a joint work between Harvard Medical School and the Dana-Farber Cancer Institute. This study proposed a visualization method to analyze cohort study data, explore patient status, and understand the possible progression patterns of diseases. To start with, what is cohort study and why we want to analyze it? Cohort study are a form of longitudinal studies that recruit patients who share a defined characteristics and follow those patients over a period of time. Cohort studies play an important role in clinical research. Those studies contribute extensive patient observations across multiple time points and offer valuable information for researchers to track patient status and understand disease progression. In our previous study, we present OncoStrat, a tool for the visualization of longitudinal clinic and cancer molecular data. OncoStrat visualizes patient cohorts as temporal heat maps and thinking diagrams, and supports the interactive exploration of a wide range of clinic and molecular features. However, the analysis of cohort studies can become increasingly challenging with extensive datasets. With a large number of observations across multiple time points, it's difficult to extract meaningful patterns. In order to improve the analysis scalability, we proposed STRAT-STATES as an extension of our previous study. We aim to address three scalability-related challenges. The first challenge is related to the number of patients. A cohort study usually includes a large number of patients. Those patients share a certain defined characteristics. For example, they have the same disease, but they may also have different gender, age, and other patient-level features. Second, each patient may have different lengths of observations, making it difficult to summarize data and extract meaningful patterns. Third, for each patient, a number of observations are made at each time point. Analyzing those observed features can be challenging, especially considering the existence of irrelevant features and the correlation between different features. To address those challenges and scale the analysis of clinical study, many algorithms have been proposed to model patient status and understand disease progressions. However, most of those algorithms are hard to interpret and require careful hyperparameter fine-tuning to generate meaningful patterns. Instead of using an end-to-end model, we employ a phase-wise method to provide users more control over the process of disease progression analysis. In the first phase, states are identified to describe different patient status using the value distribution of a set of observed features. For example, suppose three feature values A, B, C are recorded for each patient at each time point. Then we can describe different patient status using different states based on the value distribution of the three features. After state identification, common state transition patterns are summarized. Those transition patterns can help users better understand the different paths of disease progressions. Patients are further grouped based on the transition patterns to reveal potential associations between progression patterns and the patient-level features. Based on this two-phase analysis workflow, we proposed STRAT-STATES, an interactive web-based tool for state-based visual analysis of disease progression. This tool integrates existing sequence mining and clustering algorithms with a set of visualization designs to enable scalable and effective visual analysis. I will walk you through all the three views of this visualization tool and demonstrate its usage using a synthetic COVID-19 dataset. More information about the design, implementation, and other case studies are available in our paper. To start with, users need to load the data to the Strata states. We support loading through CBioPortal API or uploading from the local computer. Once the data is loaded, the visualization is updated and presents the identified states and the state transitions. The states are learned from time point features using unsupervised clustering algorithms. Users can click and add interchanged time point features, remove unwanted features, and modify the number of features as desired. Users are also allowed to merge or split those states based on the observation. Therefore, users are able to participate in the state identification process and integrate the domain knowledge, the observation about the data, with the automatic clustering algorithms to better collaborate and improve the state identification results. The state identification view consists of three visualization components. The scatter plot projects the high-dimensional time point features into a 2D space and offers users an overview of the state identification results. Here, different color indicates different states. The glyph matrix explains the characteristics of each state by showing the value distribution at different time point features. Each row is a user-selected time point feature, and each column is an identified state. The glyph in the cell represents the value distribution of the corresponding state. Since the number of records in each state can vary a lot, the commonly used histogram can make it hard to see the value distribution of states that have a small number of records. Therefore, we propose a glyph design whose x-axis indicates the number of records and whose y-axis indicates the value of the records. The new glyph design can help users observe and compare the value distribution in and across states more effectively. The area chart shows the temporal distribution of each state. The three visualization components work together and enable users to examine and interact with identified states from different aspects. For example, in this dataset, the glyph matrix shows that state A has a high occurrence frequency, which is indicated by the large width, and a low feature values, which is indicated by the low glyph height. The area chart shows that state A commonly occurs at the beginning of the disease. In the scatter plot, we can see that state A, which is indicated by the green, and state E, indicated by the orange color, are close to each other, indicating that they have similar feature value distributions. We can then examine state E in the glyph matrix view. Here, we can see that state A and state E had similar value distributions for the first three time point features. The main difference between state A and state E is lactate dehydrogenase. At this feature, state E has higher feature values than state A. The area chart shows that, unlike state A, state E commonly occurs in the middle late stage of the disease. Since the feature selection will directly influence the state identification results, we provide a feature manager to help users explore time point features. We provide a list of metrics for users to compare and rank features, and select features based on these ranking results. The identified states help users effectively summarize different patient status. But how do patients transit among those states? To answer this question, we provide a transition overview to show the patient state transitions. Those state transition patterns can be intertwining and lead to visual clutter, making it difficult to identify its insightful patterns. Therefore, we group patients based on their state transitions. For example, in this dataset, patients can form two groups with distinctive transition patterns. Since the transition is directly influenced by the state identification results, users can refine the state identification based on the observation of the state transition. For example, user may find that state A includes a large number of records and occurs both at the beginning and at the end of the disease. Therefore, users can split state A into two states. The transition is then updated and reflects this modification. Meanwhile, to better understand the patterns in each patient group, we present those patterns in an interactive table to facilitate the analysis and exploration. Users can rank those patterns based on their frequency and search interested patterns. More importantly, those patient groups may have distinctive patient features representing the correlation between the state transition and the patient features. For example, in this synthetic dataset, we found that patient group 2 has less female patients, more older patients, and a very low survival rate. More details about the state transition can be then investigated in the detailed view. For example, user can examine the medications the patients received. To facilitate the analysis, users can also combine event features. For example, here, users can combine features that are related to the same medicine. This helps us to identify the correlations between different event features, as well as the relations between the states and the state transitions. To conclude, we present an interactive visual analysis of cohort start data through state identification and state transition summarization. We design and develop a web-based open source visualization tool, ThreadStates, to support the two-phase interactive visual analysis. That's all. Thanks so much for listening. You can find more information at our homepage and our paper.